April Contreras, Department of Psychological and Brain Sciences, Advisor Rochelle Hines and Dustin Hines. The Psychedelic Renaissance, Reviving Hallucinogens as Therapeutics. April Contreras is a third-year PhD student in the Interdisciplinary Neuroscience program. She uses animal behavior and electroencephalography to explore atypical modulation of the cortex with the goal of developing novel therapeutics for psychiatric and neurodevelopmental disorders. April is a champion of promoting access to graduate education, the sciences, and the outdoors. She is an OUMP mentor, coordinator for the Hines Group STEM Outreach Program, and founder of the UNLV Mountain Club.
It is 2022, and we are living during an exciting period of rebirth and innovation. Scientists and clinicians are embarking on a mind-altering psychedelic renaissance. Today, we'll take a step back in time and through history to understand why there is a renewed interest in psychedelics as therapeutics and the promise they have for transforming the treatment of psychiatric disorders. Entheogenic or mind-altering compounds can be found in many different cactus and fungi, including the San Pedro cactus, peyote cactus, and psilocybin mushroom, better known as the magic mushroom. Take a hike through the Nevada desert or even a stroll around campus, and you're likely to stumble across these plants without even realizing it. Psychedelic plants have been used expertly as medicines and in rituals by shamans since pre-Columbian times. Mesoamerican cultures recognized the utility of entheogens, and evidence of this can be found in Aztec statues adorned with mushroom caps and other psychedelic plants. Villages in these regions continue these practices even today. But if psychedelics have been used to heal throughout history, how do we still know so little about them? And why are they just now being considered as viable treatments for psychiatric disorders? Psychedelics were labeled as harmful and without therapeutic benefit in the 1960s. When they were banned, psychedelic research was halted and stigma surrounding their use grew. In parallel, the number of individuals afflicted with a mental health disorder has increased, and current treatments are not effective enough. But a combination of recent changes in beliefs surrounding psychedelic use, as well as the pressing need for novel therapies, has reignited research interest in psychedelics, and modern science has a lot of catching up to do. My research begins to unravel new questions about how psychedelics change the brain, with the goal of revealing how these changes promote fast, long-lasting therapeutic effects. To accomplish this, I examined both behavior and brain activity in mice. And now I can't just ask mice what they see during a psychedelic trip. So I first started by using a validated measure of psychedelic action in rodents called the head twitch response. This is a subtle movement of the head, which is a lot like the twitches that humans make on a regular basis. The head twitch response in mice only occurs upon application of serotonin neurotransmitter, or brain chemical, shown in the graph on the left, and hallucinogenic compounds which act on serotonin receptors. In my study, I found that a potent synthetic psychedelic doesn't just evoke the head twitch. It also causes the animal to stop in its tracks, gazing into the distance. I then quantified the time between each stop and head twitch shown in the graph on the right, and found that they occur one after the other with just seconds in between them. Next, I wanted to know what was happening inside the brain. We can read brain activity as brain waves using a technique called electroencephalography, or EEG for short. And the speed and size of these waves can tell us a lot about the brain state of an animal or human. After psychedelic treatment, I found two very distinct brainwaves that always occurred when the animal stopped in its tracks, phase 1, and then had a head twitch, phase 2. These waveforms were fascinating because they, for the first time, provide us with a window into the brain to understand hallucinogenic activity. Even more surprising, the time between these two waveforms and the behaviors that I saw quantified earlier were the same. There was no difference in the coupling of these waveforms and the stops and head twitch response. This indicates that these distinct correlates can be informative about how the brain changes immediately following psychedelic treatment, and for the first time provide us with behavioral and EEG markers that we can manipulate to better understand therapeutic effects. This work has major implications for treating psychiatric disorders ranging from major depression to substance use. In revisiting millennia-old medicines, we can pave the way for innovative psychiatric treatments. The psychedelic renaissance is in full swing, with so many questions that I'm excited to answer. Thank you.